So this is an extension of the monohybrid cross video. We're going to take a look at monohybrid crosses as they pertain to genetic disorders, which are essentially disorders that are caused by, well, defects in your genes. So there might be some kind of mutation, which means change in your DNA, those little letters, those nucleotides. There's a change where there shouldn't be, and that leads to a gene product, like a protein or something that's supposed to be made in your body that's not made properly, which can lead to a disease. So the readings for this section can be found on page 204 to 207, and to check your understanding, you can do questions 1 to 6 on page 207. So we're going to first start off by talking about something called recessive disorders, which are disorders that require you to have two recessive alleles to have them. So if there's a disorder that's recessive, to have the disease you need to be little a. But if you have the dominant allele, then you'll be healthy. So you'll be healthy if you're big A, homozygous dominant. You'll also be healthy if you're big A, little a. But if you don't have any of these healthy dominant alleles, and you have two of the recessive alleles, then you're going to have the condition. Now, the heterozygous individual we refer to as a carrier of the disease. A person is healthy, they don't have the disease, but they carry the recessive allele for the disease with them. So that's how we call them a carrier, because they carry that recessive allele with them. The person who has the disease is not a carrier, they have the disease, they're little a. But the heterozygous individual is a carrier because they don't have the disease, they don't have the symptoms of the disease because they have the healthy dominant allele, but they do carry that recessive allele with them, which can be passed on to the kids. So even though somebody might not have a disease, they can still pass on the disease to a kid if they mate with a person who also is a carrier. So let's take a look at this picture over here. We have a carrier mother and a carrier father. The disease is represented by this pink color here. And then so let's call that little a. And the healthy allele is big A over here, big A over here. So these are heterozygous individuals. Both these parents are carriers. They don't have the disease, but they carry that sick allele, the recessive allele that makes the kid sick. So it's hidden in them. They might not even know it's hidden in them. And so they have some kids. And then what they realize is that, okay, if they both pass on big A, then their kid will be healthy. If one of them passes on big A, so let's say the father passes along big A and the mother passes on little a, the kid will still be healthy, but will be a carrier, so that wouldn't even that would not affect them. And then same thing could happen here. If the mother gives big A but the father gives little a, you could have a heterozygous individual, which would be a carrier. But if both parents pass on little a, then you have a child who's affected by the disease. So the parents may have never known that they were carriers, and then they end up having a kid with a recessive disorder, and that's how they realize, okay, they must have both been carriers of the disease because their kid got little a. And so in this video, we're going to take a look at how to do Punnett squares and predict whether kids will have diseases or not using this idea of carrier, heterozygous, and homozygous dominant for diseases. This is no different from the monohybrid questions we saw earlier. It's just now we're dealing with diseases as the traits or characters instead of eye color and hair color and widow's peak and all that. So this is one example of a recessive genetic disorder called cystic fibrosis. And you can see that this disease leads to a significant reduction in your life expectancy. So people with cystic fibrosis, we'll call it CF, live for less than 40 years. And usually what happens is there's a thick, sticky mucus that's produced in your lungs, and you have to drain those lungs. So here you can see somebody trying to pat the person on the back to release or loosen the mucus in the lungs so that they could spit it out of their body so they don't choke on it. And that usually leads to a lot of infections. Oftentimes when they have that sticky mucus, and there's blockage, and there's also some enzymes that should be released by the pancreas, but they're blocked when you have this disease. So cystic fibrosis, again, is a recessive genetic disorder, and usually it does reduce your life expectancy quite a bit. So what I expect you to do is when you go through the readings to pay attention to those different disorders, know which ones, know some recessive ones, know some dominant ones, and be able to give some examples of symptoms that it might cause. And I'll show you more of what I mean in just a minute in the table. Before we do that, let's take a look at the cystic fibrosis sample problem. So here we have a carrier testing that happened. So carrier testing is basically where somebody gets tested to see if they have a recessive allele for a genetic disorder. So they might be healthy, but before they have a kid, they do what's called genetic screening. And that's when you test for what alleles you have. And so parents might go and say, okay, we're both healthy. We want to see if we have any recessive alleles for recessive disorders in us. Are we carriers? And so if you do that, you can figure out what are your chances of having a kid with a certain disease if it's recessive. So this couple went for carrier testing and genetic screening, and they found that they are both carriers. They're both carriers for cystic fibrosis. And so here we have a series of questions that we're going to go through to answer based on that scenario. I'm going to do it on the next slide so there's more room to write. You should do this along with me. Try it yourself. Pause the video while you're trying it, then come back and see what my answer is to compare. So as part of a carrier testing, the parents go through genetic screening and they find that they're both carriers. What's the phenotype of mom? What's the phenotype of dad? So they're both carriers. Remember that carriers means that they're healthy. They don't have the disease, so we're going to go with the healthy phenotype. If they had the disease, we'd say, well, they have the cystic fibrosis phenotype or the disease phenotype. So they're healthy because they're carriers. What's the genotype? Well, that requires you to know what a carrier means, and that means for a recessive disorder, a carrier is big A, little a. For both of them, big A, little a. What are the chances that they have a child who is unaffected by the disease? What are the chances that they have a child who is affected by the disease? What are the chances that they have a child who is unaffected, but is a carrier? I should have said but is a carrier. And then what are the chances that they have a child who only has healthy alleles? And then we're going to support our answers with Punnett squares. So let's first write out what our cross is. We have a parent that's a carrier, another parent that's a carrier, so mom and dad. We'll make our Punnett square. And we'll put our gamete alleles at the top and side, so big A, little a, big A, little a. Now, remember, this is a monohybrid cross with heterozygous individuals. You could have already answered this question without a Punnett square if I hadn't asked you for one. Hopefully you would have known that it's a 3-to-1 ratio, 3 dominant to 1, right? So just to show you how I would have solved that without a Punnett square, I would have had 3 healthy, I'm going to put H for healthy, to 1 disease, D for disease. Because disease is my recessive, healthy is my dominant. So I would have had 3 out of 4 chances to have a kid who's not affected by the disease. And I would have had 1 of 4 chances to have a kid who is affected by the disease. For the last 2 questions, you could have also figured it out using your genotypic ratio, your 1 big A for every 2 big A, little a, for every 1 little a. So in E, we had, what are the chances of a child that we have a child who is unaffected but is a carrier? Well, if the child's a carrier, you have 2 out of 4 chances to have a carrier. What are the chances of having a child who only has healthy alleles? Well, that would be 1 out of 4 chances. So you could have solved that using your ratios. In a multiple choice, that would be extremely useful, so it would save you a lot of time and you wouldn't have to draw Punnett squares. But in this case, we are asking you for a Punnett square. So let's go and do the fertilization events in the squares here. So we have big A, big A, big A, little a, big A, little a again, and little a, little a. I just once again want to emphasize why there's 2 big A, little a's. Remember that there's 2 ways to get the heterozygous. Let's say this is mom over here, and this is dad over here. The first way is that, let's make this top one mom, and this side one here dad. Mom could give little a, and dad could give big A. That's one way to make the heterozygous individual. Or the other way that could have happened is that mom could have given big A, and dad could have given little a. So sometimes we represent this as, let's say if this is dad, little sperm, and this is the egg over here. And that's why there's 2 ways to get the heterozygous individual. There's only one way to get the homozygous dominant. Mom and dad both give A. There's only one way to get the homozygous recessive. Mom and dad both give little a, little a. So that's why there's two ways to get the heterozygous individual. So let's go answer our questions. What are the chances that they have a child who is unaffected by the disease? And so to be unaffected, you have to have that big A. And so there's 3 out of 4 chances. 3 out of 4 chances that we have a child who's unaffected by the disease. The chance of having a child who is affected, well, little a, little a. Chance of having a child who is unaffected but is a carrier, well, we have big A, little a, so that's going to be 2 out of 4 chances, or 1/2. And here, sorry, I didn't put the chance down. The chance is 1/4 for the disease. So always reduce your fraction when you can. What are the chances they have a child who only has healthy alleles? Who only has healthy alleles. So not who has healthy alleles, but who only has those healthy alleles. Who has healthy alleles only, I should say. And so that would be the big A, which is 1 out of 4, just like you did with any other monohybrid questions. Or I could ask you as a percentage, 25%. So we can see here the similar example that was done with cystic fibrosis. You have your carrier parents, and you can see what your chances are of having kids that do not express cystic fibrosis. 75% chance, because you have 3 out of 4 chances that your kids do not express cystic fibrosis. So this is the table of disorders I was discussing earlier. It is important that you do know some of these recessive disorders. I would know at least 2 recessive disorders, so if I ask you for examples, you can discuss some recessive disorders and tell me some of their effects, whichever one you find more interesting. So you'll notice that a lot of these diseases are caused by recessive alleles. And so if they're caused by recessive alleles, you need to have both recessive alleles in order to have the disease. Some of the diseases you'll notice are dominant. They're actually dominant alleles that cause them. So you only need one of the dominant allele to have the disease. Big A, little a, that would give you the disease. Or if you have both, you'll also have the disease as well. That's why you can't really be a carrier for a dominant allele disease, because as soon as you have the dominant allele, you have the disease. You don't just carry it, you have the disease. Whereas for the recessive one, if you have big A, little a, you're healthy, but you do carry the allele for the disease. Now, I want you to notice that a lot of these diseases are recessive, and there's fewer that are dominant. I want you to think about why in populations it's actually easier to pass on recessive alleles than dominant alleles, why they can stay in the populations for longer periods of time, as opposed to dominant, which don't stay in there as long. Think about why that is. If you do understand the concept of recessive and dominant, then you'll have a pretty good idea as to why that is and what your explanation should be. What I would do is I would pause the video. Pause the video, don't just wait for my answer, try to come up with your own answer, and I will give you a general idea once you've done that. So pause the video, write down an answer, discuss it with someone if you're watching the video with them, and then I'll discuss my answer in a few minutes. So if you think about it, a recessive allele is hidden in the heterozygous individual. And so this individual is healthy throughout their life, and they can reproduce and pass on their genes. And they could keep passing on their genes to their kids, and some of their kids will end up being carriers, and those carriers can pass on the genes, but the point is that this recessive diseased allele will stay in the population. Yes, some of the kids might end up sick, but you will still have some of the kids that are carriers. So these kids may not reproduce, or they might die early, sadly, and that knocks their alleles out of the population. But you still have these carriers that hide that diseased alleles with them, and they reproduce and pass on the genes. Whereas if you have a dominant allele disorder, as soon as you have that dominant allele, you have the disease, and you might not be able to reproduce, or you might die young, and so you're not passing on that allele anymore. So it's not hidden in you. It's shown right away, or usually at an earlier age. Not all the time. Sometimes some of the diseases might express themselves at a later age and after you've reproduced. But the idea is that we do see these a little bit earlier, and so the individual may not reproduce, may die before reproductive age, and that's why those dominant alleles aren't passed on as often as the recessive alleles. Again, I do want you to know at least 2 recessive disorders and at least 2 dominant disorders, along with the health effects that go along with them. And you should be able to do Punnett squares with genetic disorders.